Okay, so we talk about like why protocol is important in last video. Like protocol is just important to guide you through experiment, help you like help you mind those details and for your safety and efficiency. And now we're going to talk about what exactly a protocol is made of. So, if you want an example of like a really detailed example of a protocol, you can just go through my notebook on my desk, and I can send you like once I can send you a protocol in your training. Pretty much later on, there's a like detailed example of protocol. But here is like you start with your notebook code. Like when we say that notebook is connected to everything's like linked to everything in your experiment. So like just first decide on what is your notebook code. So just preserve like 2 to 4 page for every like experiment. And like what I like to do is like before I start planning the protocol, I will go to my notebook and make sure so I know what like which page exactly I'm going to start. And that is the code for this experiment. And so just preserve 2 to 4 page for every experiment, and like 1 to 2 will be for protocol, 1 to 2 for the result. And you might find out later that like your result is just taking more space than 2 to 4, but you can just like print it out and just like stable it and paste them into it as on that page as long as it is stable and will stay there. Okay, so then you will have your date like here, your date, and then your like the project and experimental name, like and your objective, like it doesn't have to be long, but it has to have the main points of why you're doing it and what questions you're asking. And like remember the objective before the experiment is super important because it guides you in your data analysis. So also like you can plan on what are you going to do to analyze data, like saying that you're going to run the data through pigs to identify the protein, and then run through deuterative for rate analysis, or run through CHAP for fold like stability analysis. So just like make sure you mention the question you're asking in your objective, because that is important to guide you in your data analysis. Okay, so the next thing is about what are you going to use. So here is, so first like, and also like safety. So like you will have safety caution, like precautions, like cautions, like what kind of PPE you're going to wear, like what kind of container that will compatible with your solvent, or like do you need to work in hood, and just like if there's like if it is human samples, it will mention that human sample you have to work in the hood, like biosafety hood. So like this part is what you are going to use, your material. So just like plan like really clear and for detail that what you're going to what like plan what you're going to have. So first, for of course, we are biochemists. We study biological difference in between samples. So are you going to use any biological samples? If you are, like make sure you mention like what are the source, and like here like for the samples this is using a serum samples. Like here you have like the sample name of this human, and then it tells you it is humans, and we make like we have the protein concentrations BCA assay from the notebook LL1116. So the detail you're going to have here, and then it has the protein concentrations, well, how are you going to dilute it, how much you will have, and then it also have a key of sample labeling of like, and because you're not going to write every detail, like there's just not enough space for you to write all the details in the vials on the vial on the tube. So you give it a little bit, simplify it. Like here in this examples, we have 4 treatment. Diflunisal is a drug, and we have the different concentration treat to the tissue source, and then for each treatment we have different concentration of guanidine. So that is like this row. So and that makes us, we have like 36 samples, and our sample coding is like A refers to like 0 micromolar of treatments, and A 20 like 225 is we have like it is denatured at 2.25 molar of hydrogen, like guanidine hydrochloride. So you have a key of your simplified versions of your labeling. So that take care of like the biological samples. Then like you will know exactly what are you analyzing, like what, like if when it comes to your data, you know what they are and how you're going to compare them. And then since we're biochemists, the next thing is the chemical reagent we're going to use. Like here list all the reagent you have, that is like your like stock concentrations. And also you will have a recipe of recipe of how to make it. So like as I mentioned, we want to do all the calculation before we get into the lab. So here for the guanidine PBS, it tells you like how much guanidine you're going to dissolve in this like 45 ml of like solvent, and this is like what solvent you're going to use. So make sure you have a recipe, and that really make your life a lot easier. If you want to, you can even like write down like like the calculation step, how you get to your end, like your concentration here. Just if somebody want to check your calculation, not check your calculation. If somebody later want to learn about how you get to this conclusion, they have the idea. And then also like just like make sure if you like put on the manufacturers of the uh, manufacturers and the cat number of the chemical. So if you want to order the same thing later, you have those reference already. And also just when it comes to time, maybe like when you're troubleshooting that you have no idea what's going wrong, and then like you will, I hope that is not going to happen to any of you, but like sometimes you want to troubleshoot like your reagent, is there's like a problem with reagent that causing your problem. So just a story against here. And one of my professors, that's like from one of my biochemist professors, and then he said like he was working in one university, and then he has really successful result. And then he was looking for the synthesis of the plants. He has really successful result, and that then he moved, like he changed job to another university and different city, different state. And then he tried, like he has all the details, and then he will try to reproduce his result, like his experiment in the new university, but he can never get the same things. And it is really frustrating. Like you will get to know that like if you cannot reproduce it, it is like you cannot say that is real. So you get really frustrated, and he then like look at like exactly how the two set of experience are different, and they boil down to the point the only difference is the spinach, the veg spinach, like the green stuff, green veg that is supplied from different supply, because they always go like get those spinach locally. So apparently some mineral or the soil provide different nutrition. That is their variable. So it is like when it comes to the point you want to troubleshoot your reagent and your material, it's important that you have noted like, okay, what manufacturers and what category that you were using. So, and also it's easy for you to know like to know like where you're going to order it. And also like it is not here like not in this column, but like it is really useful for you if you can add the columns to it. It's like, okay, where are you going to, like where the chemicals was stored? Then you wouldn't like, if somebody want to follow your protocol, they don't need to hunt for the reagents in the lab. And you can notice like it also gives you some condition of the solvent, like how they can be stored, and like not exactly all the detail, like as we mentioned, like you also like for the reagent you also need to know like what is this container they can be safely stored at. Okay, So here, so and also like you are going to like list some of the equipment you're going to use, like here, like we're going to use LCMS or Orbitrap, and like it did not list all the detail of equipment because we have like for this one we have been like doing it a lot of time, but for something that is new, it is worth to say, okay, we're going to use Apple door tube or like span filter, and sometimes you like refers to older protocols, then okay, I can refer to that. So in just like the ideas, you want to include all the detail, as much as detail, and then somebody you can reproduce it really easily, and somebody else can reproduce it from what you do easily. Okay, so um okay, so I'm just next one. I'm just going to show you a uh, let's go, jump up here a little bit, and I want to show you the inventory. So I already gave you guys the link there here that can lead you to the inventory. And then now I'm just going to show you how it looks like. Okay, here it is. Well, first, this is a housekeeping assignment that will be updated every semester. And this is a label, a map of like what is each drawer's name, like the section A, B, and C, and D is on the other side. Okay, so here I have new sheets, like two sheets tell you about like where the chemicals are stored. If you ever want to look for some chemical, you can come here and search for that. And we'll talk about if you're the one intake the chemical, when somebody like deliver the chemical, you're also going to log it onto here. Okay, in the web, like the computer is connected to internet, so you can do the inventory, like log into the inventory there. And then we also have like some supplies and equipment, like it tells you the address of the drawer and what it is. So if you ever want to look for something, you can look for it there. Okay, so that is the inventory. Okay, so let me know if you have more questions. Like that is like you should all have access to that already. Just examples like this is like, oh, just thanks for Anisha that she did amazing job on like taking inventory of what we have in the lab here, where we have clear labels of what is the content in the drawer and the address of the drawer. Okay, so let's go back to making protocol. So then the next thing is like the method. You're going to step the method out, like just make sure like you can follow it stepwise. It doesn't matter how many steps you have as long as it is clear. And then if if you modify anything from the project like previous protocol, make sure that you have at the end section you mention like why you make that modifications, and then also like if you are doing like a reference like we are modifying from some publications method, then we also reference it at some place at the end of the note session of this protocol. Okay, so like here, if it's a not new project, just include as much as detail and of why we're doing it. If there's an obvious why in front of paper, then just reference it into that paper. So there's some examples like you have like the calculation of like why you're going to like the mole ratio of your reagent and how it is connected to like Cleodotin of the protein. And this is like folding like a labeling experiment. You like add iodine and incubate at room temperature and then quench it and then incubate again, and then add another 6 molar of quantity just to denature the protein. And this is a diflunisal treatment. Like just the point is like we have them all step out, including the volume, what you're going to do, and for the time and temperature. And it is really beneficial for people to know all the setup of your experiment. Okay, so that is what you're going to have in the protocol, like just for the protocol making. So like there's a lot of information, but don't worry, just as you go, as you practice, you will start to internalize those. Okay, and the next step, okay, so that is protocol making. Now we are going to talk about labeling. Okay, so we have notebook code, and the second thing, labeling is super important because you don't want to lose your samples, especially your biological samples. Those are the samples that like the I will say that's the most valuable assets in in our lab, because those are something that we cannot use money to redo it if lost. And also raw data, mass spec raw data is also super important. But here is just like why we need to labeling, just as long as it is clear and we need to storage at the right place. So here is some of the examples of how you are going to label your sample. First of all, label cap and both sides, because like just I want to emphasize, because you're going to store your samples in a freezer box, and in that freezer box, I will show the picture later, it is really like just everything stored vertical, and it will be super beneficial if you have labeled the cap, and so it will help you easier to find your samples. You don't have to like pull up the sample on the side every time. Okay, so here's for biological samples that is stock long-term, like stocks biological sample that will be stored long-term, like in -20, -80, like for years, that you want to have notebook code, sample name, and the concentrations. Okay, and for those chemical reagents that, and like for long, like when I say long-term, that is like maybe like couple months, couple weeks, a couple months. And because if it is really long-term storage, you'll have to have secondary label from the stockroom. Okay, but you have to have your content, like here, like it is urea, concentration is 8 molar. This one we did not pH, so there's no pH here, and it has a date of made, and it's important because you want to know where you're going to discard it. Okay, also your it shows that you climb your assets, you're probably, but also important because if somebody see it left on bench, he knows where to return. And if you have reagent that is on bench that is not labeled, is not clear, and is out of date, it will be guaranteed to discard by the cleaning people. So if something pressure, make sure you label it clear and store it well, and just make sure you store it like proper container, and we're going to talk about it in a bit. If it is experimental samples like that is temporary storage, like now it's on your bench that you're doing a bench work, after it's in the media product, like those filter, just make sure you have your notebook code, the sample's name, and then you have the reagent name of concentration, just like, and like notebook code tells everybody you're working on it, and they will not just throw it away if they see it is left on bench. And also just like you will not confuse yourself when you're doing your preparation experiment. Just one note, like I learned it hard on my experiment, try to keep only one reagent, the reagent that you're actively use at that time in your bench area. Like if it is like on the cleaning, like a solvent pad. So the reason is that will not confuse you. So here's another story. So I supposed to put ABC ammonium bicarbonate into my protein digestion, but instead I put and the other, like the next day after incubation, I see like, oh gosh, why it is all crystal. And then apparently I did not put ammonium bicarbonate. I put 6 molar quantity hydrochloride, and 6 molar quantity hydrochloride is a salt. 6 molar is really high concentrations. And so what happened is after incubation at 37 degree, it just evaporate, all the solvent are evaporate. So it's like all crystal there, and of course the protein digestion did not work. And what happened is I count like there's a ammonium bicarbonate quantity just like in the bench area, and I just confused myself. I did not read the label well, which you should read the label top and side 3 times before you use your reagent. And also just what I learned is like if you only have one active reagent on your sim in front of you, it is unlikely you'll mess up. Okay, so that's important about label and reading label. Next slide, we're going to talk about sample storage. As I mentioned, that it is important to store them because the biological sample is the assets that we will not be able to recover if it is lost or contaminated. So first, have like clear label, and always keep them on ice when they are not in fridge or freezer. Temperature is important and it will ruin proteins. Just make sure that, and return them to the free -20 or -80 after you use, especially your stock samples. But if accident happens, report it honestly so we can move forward properly. Just like as we said, like we want to know it is important, but we are human. We make mistake. If mistake happen, let's just report it. And in the same time, be really cautious not to make mistake. That if mistake happen, let's talk about it so we know how to move forward. Okay, so it is like why you want to have the cap label, because it helps you recognize them in the freezer box, and you're going to organize your samples in your freezer box, and you will be giving one once you get to the point you have samples that you need to store it, and just label the box clear as well. Okay, and this is like in the process of your work, you have so many samples like lining up together. You have to have assistant to make sure you don't confuse them. Okay, well, I love stories, and I bet I think I already shared a couple of stories here. But here's the slides. So JC's birthday, and then we are that years we decided to surprise him, telling him that the -80 was not working and smoking. We had dry ice, put it on the floor, and the room was dark. And we also even have a beeping sound, like alarm from the cell phone play. So when he walked, like somebody go in, like went in for him that the -80 is not working. So let's stop here again. Like why -80 is so important? Because that is all our like mouse experiments, like all our mouse experiment teachers are there, those like the stock of stock, like we don't want to mess that up. So somebody, like we decided to surprise him as that -80 is not working. So like of course he was so worried, and he like run to the lab and checking on the -80, and like even though he saw like there's a happy birthday sign hanging on the door, like he's still worried about it, like just checking everything. And then we surprised him with happy birthday. So yes, it was a good surprise, and they just tell you like how much -80 and sample storage are important. So after that day, he immediately ordered the second -80 that is in our web lab that you see like the green one. So we want to store the like we really want to keep our samples safe. Okay, the other thing is about like sample another story about like Mars colons. That's the reagent is to be kept in the fridge, but accidentally it was put on the bench for 3 days over the weekend. So gladly my student like told me that it happens, and it is good that we know so we can troubleshoot and like to learn if there's any bacteria growth in that over the weekend, or like can we still like purify, pull down the protein as it was. So it is important, if accident happens, just let us know and we can figure out from there. Okay, and then so talking about sample storage, like we talk about biological sample storage, like they need to be on ice. Chemical storage is also important because you really don't want to have contamination in your buffer. So we need to know like how we're going, like what temperature we're going to store them at, like what container, something. Most of the chemical we store is at room temperature, like what container we want to use. Like for some of them, like organic solvent, we want to store them in glass vial, and we also want to know the expiration time. That is why we have the date of make there. Okay, on that line, I just want to know like just examples iodine like in folding group that we use is suppose it cannot store in glass because plastic absorb iodine, so you have to store in iod like glass vial and it have to be kept from light. So we have a foil folding like covering the glass vial. Okay, and you also want to mind the lid that you're using. So if you're using a small glass vial that is like 20 ml, can hold 20 ml, that is one in one of the drawer, make sure you use a Teflon lid because the lid come with it is pretty much garbage. Like just you can only like hold water, will not dissolve water, and that is a cloth like a wooden cloth here, and so it is bad if your solvent get dissolved and have those things. And guess what? We're doing mass spec experiment. Mass spec will see it. And the worst scenario, like it might like code it on the instrument and just damn it, like shorten the instrument life. So one story is this. Okay, I have a formic acid OB buffer that is like in formic acid and is a acid. And then one day I looked at like I have been using for a month and I did not like feel like notice anything more, but I have weird contamination problem in my data. I did not know what's the source. And usually I put a lid, like I usually face the lid down on a paper towel, just prevent the dust fly into it, fall into the lid. And that day, and then on that day, I opened the lid and just like, I don't know why that day, I just look at the lid and I start seeing the brown lining. And I was like, weird. It is like, should not be that. It is this part. So all the linear lining got dissolved into my solvent. No wonder I have weird contamination problem. Okay. So just like remember like what you going to use in your solvent. Like store your chemical because you really don't want to introduce contamination. That is why also temperature and if like important, if you have to put in the fridge to prevent like bacteria growth, put it in the fridge. Okay, so the last things on this slide is well, okay, let's stop here and I will do the stir technique in the next video. Stop recording.